Hello, good evening. I'd also like to thank the organizers for the invitation. It's always wonderful to come and visit Spain. I have a lot of family who live here. And thank you to Anastasia for that very interesting talk. I think one of the things that I like most about working on HIV/AIDS is the opportunity to hear from so many different areas of science. So I'm going to be talking about something completely different. And trying to convince you or tell you about some of the efforts that are ongoing and will be developed to use genome editing to provide a therapy for HIV. So just to set the stage, as you are all aware, one of the challenges of treatment for HIV is that even when people take ART long-term, we can never cure people. And this is because of the persistence of reservoirs of HIV. There is some speculation that there may be ongoing replication in drug and immune sanctuary sites, and certainly part of the reservoir is also the latent but reactivatable HIV genomes that are carried in some long-lived cells, including, for example, central memory T cells. So the idea of using gene therapy as a treatment for HIV is actually pretty old. Almost 30 years ago now, David Baltimore, Nobel laureate, first proposed the idea of using gene therapy to make cells resistant to HIV. And he called it intracellular immunization, and this is a News and Views article that was published in Science. So since David first talked about that, the field has embraced this and also moved on. Certainly, most of the efforts around gene therapy are still based on the idea of making cells resistant to HIV. But in addition, especially with the new tools that have come from genome engineering, we're now talking about using gene therapy to actually remove or disable integrated latent HIV genomes. And in addition, although I won't have time to talk about it today, we're also using genome engineering strategies to improve the immune response against HIV using strategies such as CAR T cells. So what is genome editing? So genome editing is a type of gene therapy that uses sequence-specific tools. This means that we can make precise changes to a gene. We can change both the sequence of the gene, and we can also make changes to things like epigenetic marks or impact the transcription of a gene by having an effect on the promoter or other regulatory elements. Although most applications currently are based on the use of a class of genome editing tools called targeted nucleases. So what are targeted nucleases? Well, these are basically like restriction enzymes. They make a sequence-specific DNA break. There's two broad types of targeted nuclease. At the top there, you'll see the protein-based nucleases, for example, zinc finger nucleases and talins. And these bind to specific DNA sequences because they have DNA binding protein domains. And these are linked to a non-specific nuclease. At the bottom, you'll see probably the more famous class of genome engineering tools, which is the CRISPR-Cas9 system. And this has really set the field on fire because of the ease with which you can use CRISPR-Cas9, because its ability to recognize the DNA sequence is conferred by a simple complementary guide RNA. And this guide RNA recruits and brings in the Cas9 nuclease to create the DNA break. So once you have a DNA break at your specific target sequence, what happens next depends on which DNA repair pathway the cell might use. So in the simplest case, and the most common case in human cells, the DNA break will be repaired by a pathway called non-homologous end joining, or NHEJ. And this pathway is very error-prone. Typically, if you think about the ends of the DNA break being stuffed back together again, and it's done in an imprecise way so that you will both lose some genetic material at the break, and also some of the DNA sequences can be duplicated, so that this leaves a scar. And if the break was in the middle of an open reading frame, the end result is frequently that you will disrupt the open reading frame and knock out the gene. There is an alternative pathway which occurs at certain stages of the cell cycle, which is based on homologous recombination. And in this case, the cell makes a very precise DNA repair. And it normally does this by copying the DNA sequence that is present in the sister chromatid. But as gene therapists, we can actually hijack this process. If we introduce what we call a homologous DNA template into the cell at the same time, this can be a plasmid or a DNA virus that contains a stretch of DNA that has homology around the region of the DNA break, then we can hijack this process, and instead the genetic material in our homology template will be copied at the DNA break. And this gives us an opportunity to introduce some slight changes. For example, we can change a base to change an amino acid to create a mutant version of a protein. So because using NHEJ gene knockout is a much simpler process, the first attempts at using genome engineering in any clinical setting relied on this process and the strategy to knock out a gene. And the obvious first target for this was CCR5. As you know, CCR5 is the major co-receptor that HIV uses to get into cells, and loss of CCR5 is, completely normal and well tolerated. 1% of the world's population have 2 copies of the so-called delta 32 allele and therefore do not express functional CCR5. And then, of course, this was also demonstrated by the very striking N-of-1 human experiment, the example of the Berlin patient, Timothy Ray Brown, who was cured of his HIV following receiving a hematopoietic stem cell transplantation from a donor who was homozygous for the delta 32 allele. So this really provided the impetus for teams to develop CCR5 knockout as a gene editing therapy. And the first clinical trials, which were pioneered by a team at the University of Pennsylvania, Pablo Tibas and Carl June, in collaboration with a biotech partner called Sangamo Biosciences, these first trials used zinc finger nucleases to disrupt the CCR5 gene, and this was applied to T cells, which were harvested from HIV-positive individuals, expanded ex vivo, and the cells were treated with the zinc finger nucleases. These were delivered either as an adenoviral vector or later as a messenger RNA. And then following expansion of the cells, some of the cells were frozen down so that there are backup products, and some of the cells were injected into the patients. And in addition, sometimes the patients received a conditioning step, the drug Cytoxan, which depletes some of the T cells in the patient and thereby makes space for the new cells to come in. There have been 5 completed and ongoing clinical trials to look at zinc finger nuclease T cells. I don't expect you to see all the detail on this slide, but it's in a review I published last year. I'm just going to summarize some of the highlights and some of the reported results that the investigators have publicly disclosed. So first of all, as with any initial trial, especially with a small group of patients, your primary concern is it safe? So what has been concluded so far is that the infusion of zinc finger nuclease treated ex vivo expanded autologous T cells is generally safe and well tolerated. Most of the patients actually saw increases in both their CD4 cells and the total T cell counts after receiving this infusion. And the CCR5 modified T cells could be detected in the patients and persisted long-term in vivo. And in addition, the Cytoxan conditioning, as expected, improved both the total numbers of T cells in the patients as well as the frequency of the CCR5 modified cells in a dose-dependent manner. There were also, I think, some tantalizing hints of efficacy. It's not my study, so I get to say that. First of all, I noticed that there are some reported effects on the latent HIV reservoir. This is showing the results from a cohort of patients who were immunological non-responders, and this is measuring the amount of HIV DNA in their T cells. And the graph shows the decrease in the reservoir for these 6 different patients. And you'll see that the decreases range from a rather small amount up to 3 logs for the patients on the far right of the graph. In addition, there was also some evidence that some of the patients were achieving viral control after treatment interruption, meaning after their ART was withdrawn. For example, in this cohort of patients, where 9 patients underwent treatment interruption, 6 of them were discovered to have controlled their HIV, meaning that the levels of HIV that the virus then settled out after the ART interruption was significantly lower than their baseline levels recorded before the patients went on ART. And in one case, which is the individual shown in this graph, his levels of virus actually fell down to undetectable levels just towards the end of the ART interruption. As part of the protocol for this clinical trial, he was put back on his ART therapy, so we don't know if he would have remained at this baseline level in the absence of ART. But some subsequent patients who have also controlled their virus, it is my understanding, have persisted in staying off ART for the duration of the study. So I found these observations to be quite promising, although again, very small numbers, very early stages. I've been part of a team that has been trying to develop a similar approach, but this time, instead of using T cells, we aim to go into the hematopoietic stem cells. And the advantages of this are expected to be twofold. Firstly, stem cells just last longer, so we have the potential to have a longer lasting, perhaps even lifetime treatment. And in addition, hematopoietic stem cells give rise to all the cells in the blood and bone marrow, therefore providing the potential to protect not just CD4 T cells, but also cells of the myeloid lineage, such as macrophages. I did this as part of a large team of scientists that were funded by the California Stem Cell Agency. And you may have heard that California can sometimes be a little against the politics of the rest of the United States. And at the time when our then president, George Bush, who I have to say looks like quite a good president now with hindsight, at the time that George Bush was banning the use of research into stem cells, especially embryonic stem cells, the good people of the state of California rose up and provided funding for stem cell research. And we were one of the lucky recipients of this unintended bounty. The other collaborators on the team include the biotech company Sangamo Biosciences and the clinicians at City of Hope Hospital, which is also in Los Angeles. They are experts in hematopoietic stem cell transplantation. Just briefly, we spent about 4 years developing a method to use zinc finger nucleases in hematopoietic stem cells. We developed a large-scale method to disrupt the CCR5 gene by electroporation of messenger RNA coding for the zinc finger nucleases. And in a series of experiments where we took those human stem cells and transplanted them into immune-deficient or humanized mice, we demonstrated that the treatment did not impact their function. We also demonstrated that the zinc finger nuclease-treated stem cells, or the mice that received them, were able to control their HIV when compared to mice that received only normal human stem cells. That's the graph on the left, where you see that all of the mice initially get infected, but the zinc finger treated stem cell mice were able to control their HIV over a number of weeks. And then in addition, when we looked at the level of CCR5 negative cells in the mice, both before and after HIV infection, we see a sharp increase in the levels of CCR5 disruption, which makes sense because we expect that HIV infection would select for the T cell progeny of those stem cells that are indeed CCR5 negative. So all that data led to the initiation of a phase 1 clinical trial. It's a pilot study. There are only 12 patients that are going to be enrolled. These are their characteristics. Importantly, as I think you'll all understand, we screen to make sure that the patients are not harboring any X4 tropic virus, because this is a therapy that will only work in patients who only have R5 tropic virus. And this study uses two cohorts of patients, and they will differ in terms of the level of a drug, busulfan, that they receive as a conditioning regimen. Similar to the use of Cytoxan in the T-cell trials, busulfan is expected to make space in the bone marrow so that when we return the edited hematopoietic stem cells back to the patients, they will have a better chance of going back and engrafting into their bone marrow and producing progeny cells. The clinical trial started in spring of 2016, and in October, the 6th patient was treated. I am also not involved in this clinical trial because I'm a basic scientist, but my collaborators who are doing this report that everything so far is going well, although it is still early days in the trial and they are not yet publicly talking about the findings. I'd also like to point out that in this trial, a treatment interruption is also planned about 9 to 12 months after the patients receive their transplants. And that is because it sort of takes so long for stem cells to engraft and start to produce a new immune system in a patient. So I've already hinted at some of the limitations of a therapy based on CCR5. For sure, it will have no impact against any X4 tropic virus. But I've also been thinking about what else can we use genome engineering for as a way to produce HIV-resistant cells. And especially because we are now, I was interested in whether or not we could get that other pathway to work. So again, if you remember this slide, I've already told you that non-homologous end joining and gene knockout is an easy, low-hanging fruit to go after. The more interesting pathway, the homology-directed repair pathway, is more challenging. Firstly, because it is the less frequently used repair pathway in human cells for many stages of the cell cycle. And secondly, because it requires us to introduce two components, the nuclease, but also the homology template, which makes things more complicated. My group have worked to figure out a method to introduce the homologous template, and we discovered that for hematopoietic stem cells, the optimum procedure was based on using adeno-associated virus vectors, AAV vectors, and especially those of a serotype called AAV6. The AAV virus is a small DNA virus, and we basically replace its genome with a homologous sequence from the gene we want to target. In this experiment here, we are using homology-directed repair to insert a small mutation, a restriction site in the middle of the CCR5 gene. And you'll see in the graph that as we increase the amount of the AAV homology donor, we both decrease the amount of gene disruption, the indels that we observe, as well as increasing the amount of edits. So with that technology in hand, we decided that we would attempt to engineer restriction factors. Restriction factors, which are proteins such as ApoBec3G and TRIM5-alpha, are intrinsic cellular defenses, proteins that recognize HIV and other retroviruses, and that inhibit specific stages of the cell cycle, and in some cases can trigger inflammatory responses and interferon responses. Despite their potency, HIV has no problem infecting us. And that's because HIV has basically figured out a workaround for each of these human versions of the restriction factors. For example, the VIF protein degrades ApoBec3G, and the VPU protein degrades tetherin. So in order to make, if you like, a better restriction factor, I think of this as a chess game. The virus is currently winning, but as a genome engineer, I wanted to know if we could make the next move. So I'm going to just show you a couple of slides about a preliminary project we are doing to edit the tetherin gene. So tetherin is a cell surface protein that blocks HIV release. It basically tethers the virus on the cell surface, as shown in that cartoon. In addition, it can promote ADCC, and when it is holding on to the virus, it also triggers NF-kappa B. Interestingly, if you take the tetherin protein from certain primates, for example, tantalus monkeys, this protein is now resistant to HIV-1 VPU, and therefore that tetherin is active against the virus. And the differences between the human and the primate tetherins that confer that activity map to the transmembrane domain of the protein. Therefore, we were able to use those amino acid sequence differences as a guide to introduce mutations into the human tetherin and identify a combination of mutations, we call it delta GI T45I, that produce a human tetherin that is both able to restrict HIV release and is completely resistant to VPU. And our goal, therefore, is to ask whether or not we can introduce those mutations into human tetherin and ask whether that can now inhibit HIV. So there are some challenges involved in this goal of editing tetherin. This cartoon here shows how we do the experiment. We have our nuclease that recognizes a sequence in the tetherin gene, and then our homology template on the AAV vector, which includes a stretch of the tetherin gene, but has the mutations we want to introduce. And over on the right-hand side, I'm showing you what the possible outcomes could be, as well as having no effect at all and having a wild-type tetherin, and having the edited tetherin that we want to achieve. One of the things we worry about is that there will still be at some frequency, and possibly as the dominant product, tetherin that has an indel in the middle of it and is therefore disrupted. And I kind of think that knocking out a gene like tetherin, when it is not known that people can live happy and healthy lives without tetherin, would not be a very good thing to do as a gene therapist. So to try and, I should just show you, this is data that convinces me that indels in tetherin would be really bad. We took a series of clones. We looked at the pattern of the insertions or deletions that were created by the zinc finger nuclease, and the vast majority of them destroyed the open reading frame, and we did indeed lose tetherin production. So can we think of a strategy around this? So what we are attempting to do is, rather than have our DNA break be close to the site where we want to introduce the changes, as is typically done, instead, we are trying to introduce the DNA break in an adjacent intron. And the idea is that in the intron, as long as we keep away from the edges of the intron where there are important signals, we may be able to tolerate some indels in this region without impacting production of the tetherin. So that the example on the bottom, the indels there, wouldn't necessarily lead to loss of tetherin. So again, just to show what this looks like, up at the top is the sequence of the exon and intron junction in tetherin, the position where we have located our new zinc finger nuclease, and down at the bottom is some analysis of the indels we get. And on the molecular level, the indels look very similar, except because they don't extend into the open reading frame in the vast majority of cases, we have very little impact on tetherin. So we think we can get away with doing this. Of course, it seems like an obvious idea now, and I was puzzled why nobody else had done this. And people said to me, that's because this won't work. Editing, you have to make the DNA break close to the site you want to edit. And if you try to do editing at a distance, it just won't work. And I said, how do you know that? And people said, Drosophila, yeast. Okay, so it shouldn't work in humans, but actually it does. So here's a simple experiment showing you that the degree of editing we get at the two mutations we want to introduce into tetherin when we use a zinc finger nuclease that is close to the sites we want to edit in the exon or at a distance in the intron. And we see that the intron-targeted zinc finger nucleases do indeed support editing. It is less efficient than when we have the zinc finger nucleases right at the edit site, but I think I can live with that reduction in efficiency, knowing that we've seen a big increase in the safety of the procedure. And we have ongoing experiments now where cells edited in this way are being put into humanized mice and are being challenged with HIV. I don't yet know if it's going to work. It's going to be interesting to see if it works, and it's going to be even more interesting to see what HIV does as its next chess move. So the last story I'd like to tell you about is how we could use zinc finger nucleases or CRISPR-Cas9 to disrupt the latent reservoirs. Because if you think about it, HIV, once it's integrated into a cell, looks like a gene. So we could possibly use non-homologous end-joining error-prone repair to also disable the HIV genome. And hopefully in this way, we could then allow people to be taken off antiretroviral therapy. So in order to do this, there are 3 stages. We need to design some HIV-specific targeted nucleases. That's the easy part. We need to test their efficacy in model systems, both in vitro and also in vivo. And then finally, we need to think about methods where we could actually deliver those nucleases to reservoir cells in vivo, and that's very difficult. First of all, showing you some data about nucleases. This is just an example of different CRISPR guide RNAs that we made at highly conserved regions across the LTR. In order to test their efficacy, we first use a cell line, JLAT cells. This is a T-cell line that contains a latent integrated HIV GFP reporter virus. And when you stimulate the cells with TNF-alpha, they respond and activate and turn green. And if you treat the cells with the nuclease and the nuclease is working, you see a decrease in that green shift as shown by the FACS plots here. So we tested our series of CRISPR guide RNAs, and actually, the majority of them don't work very well. I'm the first one to admit that to you. But we do have several, including 2 very good candidates that have a very strong effect on reactivation from latency. And this is probably not surprising because both of those CRISPRs are targeted to the SP1 binding sites. Okay, so we have our candidate CRISPRs that will recognize and potentially disable HIV, and they work well in a cell line. So now we are moving to look to see if we can see any evidence for efficacy in an animal model. And in order to do this, we developed a humanized mouse model of the latent reservoir. So first of all, our mice are very compliant. They take their drugs every day. That's because we mix them with their food and we don't give them any choice. When we start the mice on this sort of combination ART feed, their viral levels drop down to undetectable levels. And same as you see in patients, when we take the mice off this regimen, their HIV levels come back. At any time in the process, we can take the animals and we can harvest their lymphoid tissue, spleens, and lymph nodes. And for these experiments, we use a strain of HIV that has a cell surface expressed epitope tag called HA. And what that means is that any cell that is actively infected with HIV will also have the HA tag on its cell surface, and we can detect it by flow cytometry. The other trick we can do is we can take the mixture of the spleen and lymph node tissue and we can put it through a depletion step where we use antibodies and magnetic beads to remove the HA positive cells. And so what we end up with is a mixture of cells that will be both uninfected and hopefully also contain some latently infected cells, because on the outside they look the same as uninfected cells. We then take that population of cells and we treat it in 2 ways, either unstimulated or we stimulate it with antibodies against CD3 and CD28, which activates the T cells and reactivates any latent genomes. And by measuring the degree of virus production by the stimulation, we have a crude measure of the latent reservoir that has established in vivo in those mice during ART treatment. And so then we can ask whether our reagents have any effect against that. First of all, we do the easy experiment and we do it in vitro. We treat the cells with our nucleases. And we see that compared to untreated or a control nuclease, that treating with an LTR-specific nuclease significantly decreases the amount of HIV reactivation we get. Okay, so now we get to the really difficult part, and this is still a work in progress. How are we going to take these nucleases and deliver them to latently infected cells in a human, especially when only 1 in a million central memory T cells may be infected with a latent virus? So we're thinking about ways to target. We're thinking of surrogate markers on the surface of a latently infected cell that might indicate that cell could harbor a latent genome. In particular, immune checkpoint blockers like PD-1 and TIGIT. We know that the reservoir is enriched in those cells. Before we can even get there, we are asking if we can even target CD4 cells. So the final data slide I have is showing some preliminary data using a system that was developed by Christoph Buchholz in Germany. This is, I think, a very nice system. He takes lentiviral vectors, and on the outside of the vector, he puts an engineered measles virus protein, which also has attached to it a DARPin that recognizes CD4. This is kind of like an artificial ligand. And when we take those CD4 DARPin lentiviral vectors, and for this experiment, we just encapsulate a GFP expressing genome, we actually see that we can get some specific targeting. So first of all, at the top, I'm showing you what happens with these vectors when we take our humanized mouse spleen and we treat it with the vectors. And you'll see that all of the GFP is in the CD4 positive cells. And then in addition, we've now started doing experiments where we are injecting those vectors into the mice and then harvesting their spleen 5 to 8 weeks later and looking for the presence of any GFP. And although they're very faint dots and you probably can't see them in the audience, I have to tell you they are there. So we are now doing experiments where we are putting our nucleases into those vectors and repeating the experiments and asking if we can have any effect on the latent reservoir. So to summarize what I've told you, genome editing for an HIV cure. There are new technologies, they're being developed all the time. And what I think is exciting is that it's allowing us to use basic information, the things we know about HIV biology and its interaction with the host, to rationally design new therapies that were unthinkable even 5 years ago. Zinc finger nucleases that disrupt the CCR5 co-receptor are already being evaluated in clinical trials in both T cells and hematopoietic stem cells. My group is working on exploring editing other targets, specifically the restriction factors. And then finally, I've shown you that nucleases that target highly conserved regions of the HIV genome, such as the LTRs, can indeed disable the virus and prevent reactivation from latency. But I'll be the first to admit that there are considerable challenges involved in delivering such reagents in vivo. But if it wasn't hard, it wouldn't be fun, would it? So with that, I'll leave you with the acknowledgements of the people from my lab and my collaborators and our sources of funding. Thank you for attention, and I believe it's cocktail time now. Okay?